Hi, hello everyone. Welcome back to my YouTube channel, My Intuition. I'm Dr. Prasad Puthiyillam. Continuing my classes on chemistry for computer science and engineering stream, module 1, part 9, indicating the RBT level, revised Bloom's taxonomy level questions and their answers. Okay, so I have covered half of the first module in earlier videos. Now continuing as disposable sensors in the detection of biomolecules. So next video will be a disposable sensor for detection of pesticides. Okay, so disposable sensors in the detection of biomolecules. Different questions, possible questions of different levels of RBT, revised Bloom's taxonomy level. Analyze different types of biomolecules that can be detected using various sensing technologies and evaluate their respective advantages and limitations for different applications. Compare and contrast the use of different sensing materials in disposable sensors for the detection of biomolecules and provide examples of their respective advantages and limitations. Level 4 question. Develop a strategy to optimize the performances of disposable sensors for the detection of biomolecules, including considerations of sensitivity, selectivity, and response time. Another level 4 question. Evaluate the key applications of disposable sensors for the detection of biomolecules, including their potential impact on healthcare, food safety, environmental monitoring. Another level 4 question. Analyze the general sensing mechanism in disposable sensor for the detection of biomolecules, including consideration of transduction, signal amplification, and data processing. Level 4 question. Design a fabrication technique for a disposable sensor for a detection of biomolecules, including considerations of material selection, sensor architecture, manufacturing process. Another level 4 question. Evaluate the basic principles of a disposable sensor for the detection of biomolecule, including consideration of sensor design, sensing mechanism, and signal processing. Another level 4 question. Evaluate the choice of sensing material in disposable sensor for the detection of biomolecules, including consideration of material properties, specificity, and sensitivity. Level 4 question. Compare and contrast the advantages of disposable sensor for detection of biomolecule compared to traditional analytical techniques, including consideration of cost, portability, and speed. Analyze the limitations of disposable sensor for the detection of biomolecules compared to traditional analytic techniques, including consideration of accuracy, reproducibility, detection limit. So these are all the major questions of level 4. Usually an exam can face level 2 or maximum level 3 questions. So level 4 questions and I include like evaluate recent breakthroughs in the field of disposable sensor for the detection of biomolecule, including their potential impact on healthcare, food safety, and environmental monitoring. I think this question may not be asked because this is somewhat to the next level questions. But once you answer all these questions, surely you can answer all the level 3 as well as level 2 questions. All are included in this particular class video. So let us begin. Different types of biomolecules that can be detected using various sensing technology. So when we say about detection of biomolecule, which are the biomolecule which can be detected, and why we are doing this work? We can detect proteins. Proteins are very important biomolecule which play many different roles in our body. Their detection can be very important in medical diagnostics as well as drug development cases. For example, it include immunoassays which use antibodies to detect specific protein, and mass spectrometry which can detect the or identify many different proteins simultaneously. We need to detect the DNA. It is a genetic material which encodes the instruction for our life, and its detection can be important for genetic testing and disease diagnosis. Even for other applications also, we go for DNA testing to identify the criminals. And example for DNA detection method includes polymerase chain reduction (PCR), which amplifies specific DNA sequences, and a next generation sequencing, that is NGS, which can sequence a large amount of DNA in a short amount of time. RNA can also be detected. It's also a protein only. It is a type of nucleic acid that plays many important roles in gene expression and regulation. Detection can be important for studying gene expression patterns and diagnosing the diseases. Example of RNA detection in methods are reverse PCR, RT-PCR. We might have heard about RT-PCR during the COVID season, that is reverse transcription PCR, which amplifies specific RNA sequences, and RNA sequencing can be sequence large amount of RNA in a short amount of time. The small molecules. Small molecules are very important. Biomolecules include hormones, neurotransmitters, metabolites. Their detection can be important for medical diagnosis as well as the drug development. Example of such small molecules include the detection methods include chromatography, which separates and identifies different molecules in a sample, and mass spectroscopy, which can identify and quantify small molecules in a sample. Now the material used with the example of disposable sensor for detection of biosensors, we use the gold nanoparticles. Gold nanoparticles are very commonly used in biosensor due to their very high unique optical and electronic properties. It can be functionalized, it can be attached with other biomolecules such as antibodies to detect the target molecules such as proteins and DNA. Means if you can functionalize the surface of the gold nanoparticle, on the gold nanoparticle surface, if you add some functional groups, then it will be very easy to detect the biomolecules, and our gold nanoparticles are having high optical and electronic property. It can be transduced easily. Carbon nanotubes. Carbon nanotubes are highly sensitive material which can be used as a transducer in biosensor. It can also be functionalized for detection of the biomolecules. Then conductive polymers. Conductive polymers examples and at the later stage. There are a class of polymer that have a electrical conductivity. It can also be transduced in biosensor and can be functionalized also. Graphene is a highly sensitive material, can be used as a transducer. It can also be functionalized, therefore these are the material mainly used. Quantum dots, okay, whenever the fluorescence application comes, we have to use quantum dot. They are semiconductor nanocrystals which can emit fluorescent light, which can also be functionalized for the application of disposable sensors. Optimization of sensor performances of disposable sensor for the detection of biomolecules. Optimization, sensing material selection, how to the choice of the sensing material, it is very critical for the performance of the disposable sensor. It could be selective, sensitive, stable, and compatible with the target biomolecule. The properties of sensing material should be optimized to achieve the desired performance characteristics. Generally, it is saying here how to do that and all not mentioned, but it is very important criteria that selection of the material has to be selected according to the material, that biomaterial that we want to detect. Surface modifications we have to do. The required surface modification will improve the binding affinity as well as the selectivity of the sensor towards the biomolecule that we want to detect. Surface modification can be achieved by attacking some functional group or a specific biomolecule to the surface of the sensor, such as antibodies or enzymes to the sensing material. Signal amplification, because very small amount of signal will be created when we sense the biomolecules. It has to be amplified. It can be done such as using some nanomaterials or enzymes which can improvise or which will give a better result, or it will improve the result. A small change it will show as a bigger change, so that we call as the amplification. Transducer optimization. The transducer is very much important item in the every sensor. It is responsible for converting the signal of detecting the molecule or whatever the sensed material sensed. Then it has to be changed or transduced to another form of energy which is able for us to analyze or detect. Okay, so it is responsible for converting the signal generated by the interaction between the sensing material and the target biomolecule into a measurable signal. It should be optimized to achieve high sensitivity and low noise. Microfluidic designs. Microfluidics means very small amount of fluids are used, and its control we call it as microfluidics. It can be, this design, microfluidic design, can improve the performance of disposable sensor by enhancing the mass transport of the target biomolecule to the sensing material. You can also reduce the sample volume required for detection, because very small amount of volume, so sample volume required is also very less when we go for microfluidic design. Quality control is very critical in almost all the industries. Without that they cannot run also. The fabrication process should be well controlled, and the sensor should be tested and validated before its use. Some of the key applications of the disposable sensor in detection of biomolecules, like point of care diagnostics. A disposable sensor can be used for rapid and accurate detection of biomarkers in bodily fluids such as blood, saliva, urine, etc. They are particularly useful for point of care testing in resource limited settings where traditional laboratory based methods are not available. Means where we cannot take the hospital if you have to carry this small disposable sensor as the detection. So in the rural areas where, or the places where it is very difficult to reach, there and all we can use this type of disposable sensors. Environmental monitoring, disposable sensor can be used such as the heavy metal detection or polluting organic compound detection. They are very cost effective, they are portable, easy to use, making them ideal for the field testing, because environmental monitoring, we have to go there and check it or detect the changes in the environment. Therefore portable devices required, therefore disposable sensors are very much useful. Food safety disposable sensor can be used to detect some of the foodborne pathogens and toxins uh, during the production of the food, or storage of food. If any pathogens or toxins are getting added to that, that has to be detected. It's very important for our health. Drug development. Disposable sensor, it can be used to screen drug candidates for their binding affinity to target biomolecules such as receptors of for the enzymes. They are very useful for monitoring the efficiency of drugs in clinical trials. So before using in clinical trials, it has to be detected whether it is having working good condition or not. Personalized medicines. It's used to monitor the biomarkers in real time, allowing for the personalized diagnosis or treatment of disease. It can be used to monitor the progression of a chronic disease, such as the diabetes or cancer. It has to be monitored. That is personalized medicine. A person, he himself can handle and check what is the present status of the diabetes or the cancer, so that if any further medication needed, we can go and help. Every day we cannot go to hospital, that's why we need to have such devices with us. Now, general sensing mechanism in disposable sensors. Sensing mechanism is common to all disposable sensors. Okay, biomolecules are the one which is indicated here with red dot. They are the one which we want to detect, and this is the sensor sensing element. Okay, so the general sensing mechanism in disposable sensor for the detection of biomolecule, it involves the interaction between, that means this biomolecule command interact, sit on the surface of sensing element, then there will be some changes in that. The sensing element can be made of various materials such as polymers, nanoparticles, biomolecules, biological molecules. The sensing mechanism, it can be classified into two categories, that is label free detection as well as label based detection. In label free detection means the biomolecules, we are not adding anything extra to that. As such, the biomolecules, if they are in the environment which we want to detect, it will sit on the sensing element, or the sensing element will interact with that and will get the result. That is label free detection. Label based detection, usually we use a fluorescent dye. So fluorescent dye will be sprayed there, or it will be kept in the atmosphere, so that this biomolecule will be attached with the fluorescent dye, and only such biomolecules having affinity towards the fluorescent dye will be attached, other molecules will not be. Therefore it is very easy to detect, and amplification is also very easy in such cases. Therefore two types are there: label free detection as well as label based detection. Then comes the transducer. The transducer which converts, okay, once the biomolecule interact on the sensing element, when they have a interaction with the sensing element, there will be some changes in the sensing element, whether its mass will increase or decrease, or its temperature will may vary, or its optical properties may vary, or there may be some electrochemical reaction possible. All these are the energy, the conversion. Okay, so that energy converted, or that modification which is done on the sensing element, it has to be transduced, converted into other form of energy, so that it is for us to detect, for us to get the display, the reading. So construction of disposable sensor for the detection of biomolecule. Selection of biomolecule as a sensing element. I mean, it could be a protein, DNA, RNA, or any other biomolecule of interest. That is a sensing element selection. Selection of sensing platform. The sensing platform is where we have to keep that sensing element on which support. It may be, it will be based on whether it is an optical based disposable sensor, or electrochemical reaction, or mechanical principles are used. Based on that the platform will vary. Surface modifications. The surface of the sensing platform, it is modified to make it compatible with the biomolecule of interest. That means the surface, there will be some addition of the biolinkers or linker molecules, or any functionalized molecules, so functional groups are added, so that detection will be very easy. Immobilization of the biomolecule on the surface of the sensing platform. The biomolecule, it is immobilized on the modified surface of the sensing platform through various methods, such as the covalent bond formation, or physical adsorption, or affinity bonding. That means we should know the chemistry of the biomolecule which is used as a sensing element, as well as the platform, the material which is used as the supporting to the sensing element, which has to be either covalent bond formation should be there, or physical adsorption should be there, or any other bonding should be there, otherwise it will get separated. So that you have to see the chemistry behind that. Signal transduction. It is once the biomolecule it is immobilized, then the transducer is introduced there. In an electrochemical sensor, the signal transduction, it is based on the current generated due to the oxidation or reduction of the biomolecule. So in optical case, it is the changes in the light variation, or the changes in the color. So like that, signal transduction will takes place. So you have to identify the transducer based on the sensing element that we are used. Detection and analysis. The signal generated by the sensing platform, it is detected and analyzed to determine the presence and concentration of biomolecules of interest. Disposable components. Since the name itself says that it is a disposable sensor, therefore it should be very easily, it has to be removed, detached from the other any material which is supporting framework. If you have, it should be easy to remove, and it has to be very easily, it has to be replaced also. Now the choice of sensing material in disposable sensors. Conductive polymers can be used, like polypyrrole, polyaniline, polythiophene, which are the commonly used conductive polymers in disposable sensor for the detection of biomolecule. They can be functionalized. These are the properties why we are using such polymers, conducting polymers. They can be easily functionalized to provide the specific binding sites for the biomolecule, results in a changing electrical conductivity of the polymer upon biomolecular binding. Then metal nanoparticles like gold, silver, platinum are often used as a sensing material in colorimetric and the surface plasmon resonance based biosensors. And these can also be easily functionalized. That is, I mean, reason why we are using metal nanoparticles like gold, silver, and platinum. Carbon nanotubes. Carbon nanotubes are also widely used sensing material in electrochemical biosensor due to the high surface area, excellent electrical conductivity of the carbon nanotubes. It can be also be functionalized. So there is again another criteria which is required. It has to be property of functionalization should be there. Then the required biomolecule can be attached to sense the biomolecule which you want to sense. The quantum dots. The quantum dots are used whenever we have that fluorescent applications are used. That is label based detection cases. We mainly use the quantum dots, which are the semiconductor nanocrystals. These nanocrystals can be functionalized with the biomolecular binding molecule, certainly changing the fluorescence intensity upon the biomolecular binding. They are commonly used as a sensing material for in fluorescent-based biosensors. Magnetic nanoparticles. Magnetic nanoparticles such as iron oxide, hematite, macamite, etc., are used as a sensing material in magnetic biosensors. It's one of my research field. These nanoparticles can be functionalized with biomolecular binding molecules, resulting in a change in the magnetic properties of the nanoparticles upon biomolecule binding. Now, some of the advantages. After advantages, limitation will come. Then presently the new breakthroughs also will be there. So be with me. The cost effectiveness compared to the traditional method we are comparing. Compared to the traditional method, this is having a cost effectiveness. Cost effectiveness always required. Then second point, that is it gives the rapid analysis is very faster, real-time analysis possible, quick decision making can be done, and the traditional analytic technique it is a lengthy process, and we have to wait till the result comes. By the time the patient may die. So portability. Disposable sensors are small, lightweight, and portable, making them ideal for field applications or point of care testing. Portability means we are able to carry it. It is again other advantages compared to normal traditional analytical technique, in which we use bulkier instruments kept in the lab. Okay, cannot lift it. Sensitivity and selectivity is also one of the main advantages compared to the traditional method. They are highly selective and sensitive in special cases. The reduced sample volume, we need a very small amount of sample. No need to take out one full this one of blood and okay. So very small amount of sample are required for sensing. Wherever we want to sense that, it is very easy. Volume is very less required. Reduce the risk of contamination. Since we are using very less amount of the sample, contamination can also be less. Okay, it is designed, the disposable sensors are designed such a way that it has less contamination. But in traditional analytical technique, often the cleaning, sterilization equipment, because in disposable sensors just used with small amount and thrown, not thrown to the one, disposed properly. Whereas traditional method, we have to clean, sterilize. Limitations also there. It has got a very limited lifespan. Once the disposable sensor is prepared, within the given time period, it has to be used. Otherwise, it is a wastage. Okay, multiple measurement is not possible. Only once it can be used. Then it has to be disposed properly. Complexity of sample matrix. In disposable sensor, it can be less effective when the analyzing complex sample matrices. When the sample is very complex sample, a mixture of many, then it is very difficult. You have to separate it. Then traditional methods are gives a proper result. Time consuming, but proper results will be available. Will be proper results will be there if such complicated samples are given for testing. Calibration and quality control. It may require regular calibration and quality control to ensure the accuracy and reliability of the result. That is time consuming because if we have a disposable sensor, if I am not using it for longer some given time, then you have to use. Then we may have to give it back for the quality control to check whether it gives the proper result or not. Once they check, if I come immediately, if I use, that is okay. If I keep it for a little bit longer time, then it may give a false result also. Possibilities. Okay, lack of standardization. Lack of standardization means the similar type of disposable sensor will be prepared by different companies. Their standardization differences will be there. So small changes in the are always expected, but sometimes larger reading comparison, if you do larger differences are there, then that is not good. So lack of standardization is one of the limitation that we are facing in disposable sensors. Detection limit of these disposable sensors are very low compared to the traditional method. Extreme low concentration or extreme high concentration, it cannot be detected by disposable sensors. Some of the recent breakthroughs in the field of disposable sensors. What is happening nowadays with in the research field? We have developing paper-based sensor. Paper-based sensors, they have gained much attention due to their low cost, simplicity, and easy to handle. In 2020, during the COVID-19 season, paper-based sensors that developed and trial versions are done, and it gives the result within 10 minutes. So that is a very rapid result. Wearable sensors are getting ready, potential to revolutionize the healthcare by enabling continuous monitoring of biomarkers in real time. 21 developed the wearable sweat sensor, can detect multiple biomarkers including glucose, lactase, and cortisol in real time. Smartphone based sensors, everyone knows about this. We have smartphones, and newer smartphones are coming which will tell everything about our body, whether we have any changes in our body happening or not. Temperature changes to any viral attack, it will detect. In 2021, they developed a smartphone based biosensor. They means the researchers, which will detect the SARS-CoV-2 nucleocapsid protein in the saliva sample with high sensitivity and specificity. Biosensor for food safety. Biosensor for the detection of pathogens and toxins in food has the potential to improve the food safety. In 2020, researchers developed a biosensor based on gold nanoparticle that can detect Salmonella enterica, which is one of the food sample with a high sensitivity and selectivity. Nanomaterial based sensors are getting ready. They are having very unique properties for disposable sensors. In 2021, graphene oxide based biosensors, they developed which can detect the HCG, that is chronic gonadotropin, a biomarker for the pregnancy with high sensitivity and specificity. So that's about disposable sensor for detection of biomolecules. So please do subscribe my YouTube channel and support me for my effort that I am taking towards developing the study material. So my channel name is My Intuition 4865. Thank you very much.